targeting aging biology is possible, and we have some examples where we know it works in mice. We're pretty sure it'll work at least to some extent in people, and we think that we should try that. And if it works, here's the expected impact that we might have on risk of death from COVID-19. And in that Hill article, I did some sort of back of the envelope calculations. And basically, if you take what we know a drug like rapamycin can do in mice, and you say, okay, let's say it works half as well in people, what you essentially do is you basically push the risk of death from COVID-19 back 20 years. So, we know that if you go from 40 to 60, your risk of dying from COVID-19 goes up something like 15 or 20-fold. Basically, you reduce the risk of dying by 15 or 20-fold. COVID-19, which takes us from, a global pandemic to a mild flu season in terms of number of deaths. It's still a lot of deaths, but it's not this sort of situation where the entire world is gripped with fear and economies are being devastated.
Today's show is brought to you by our friends over at KetoBrick, the makers of one of the best tasting and most shelf-stable ketogenic meal replacements that's out there. I like this and recommend this to my clients for many different applications. For example, when you travel. If you go to the in-laws and they're not on a keto diet, or you have no idea what kind of food they're going to be offering to you, well, need not worry. Bring a KetoBrick with you and just ask for some lean protein. Or if you're on a business trip and your business partners are not into the keto thing and you're not really sure if they want to go to like a pasta dinner type restaurant, you can have half a KetoBrick and then just ask for some chicken breast or some ground beef and you're good to go. Your calories are covered. Another situation, here's how we use this a lot in our household, is when we're in the backcountry, for example, for hiking, hunting, we're doing a few day trip, I'll bring KetoBricks with me with some beef jerky or other dehydrated animal type products to get my protein needs, and you're good to go. So you can hop on over to our friend's website over at ketobrick.com and definitely check out. They have a bunch of different flavors. My favorite is the cookies and cream. It tastes phenomenal. Again, it's easy to travel with and the macros are perfect. You're getting whole real food from coconut, MCT, small amount of pea protein, and virtually zero carbs. So again, that URL one more time is ketobrick.com.
Yeah, there was so much fear, right? So if you mention anything to the contrary or just question anything about, like the fact that, someone like your mom who was sick, if they needed to get a procedure that they were put on hold for months and the unintended consequences.
Yeah, and I think that, obviously has extended now to where it's been politicized quite a bit. And so, I think that it is, again, if you were to take a purely logic-based cost-benefit analysis, I think a lot of what we have done as a society has not been the most efficient way to handle this situation. But the reality is that, this impacts individuals in different and in some cases very severe ways, and there's a lot of fear that goes along with it. And so you can't, from a policy maker perspective, take that sort of purely, we want to handle this in the most efficient and effective way, because that's inconsistent with the level of fear that people were experiencing and in many cases still are experiencing.
Yeah, that's super interesting. At what point when you were looking at the data, did you realize age was one of the main risk factors?
It was immediate. I mean, and in some ways, again, it's kind of obvious. So first of all, we know that almost all infectious diseases in developed countries have the same age-associated risk profile as Alzheimer's disease, diabetes, heart disease, most cancers, right? So if you just look at the relative risk as a function of age, it's almost identical. Influenza is a good example. So from that perspective, that was the default prediction for any new virus that came along that was highly prevalent, is that there would be a strong age-related component. And we can talk about the biology for why that is.
Sure.
It's pretty well understood, but it was absolutely what I think anybody who understands the biology of aging would have predicted. And then, of course, in our situation here, the first major outbreak was in a nursing home in Kirkland, right?
Just a mile away from here,
right.
Yeah.
So that, immediately in my mind said, oh yeah, well, that's exactly what you would expect, given that we anticipate that the most severe outcomes are going to be in older people. So I should clarify a little bit.
Sure.
It's not necessarily the case, and in fact, in COVID-19, I don't think there's any evidence to suggest that your risk of infection is influenced by age. But the risk that once infected, you will have a severe outcome is where that sort of age-associated exponential increase occurs. And I think that's exactly what we've seen, as we've started to get more and more information about closer to a true number of cases, which we still don't know, but closer to a true estimate of the number of people who've been infected, and then of those people who've been infected, how many of them had severe outcomes or died. And again, it matches exactly what we would predict for an age-related disease like influenza or like Alzheimer's disease.
And so as a geroscientist, I mean, you must have been just really excited because now obviously everyone wants to, they see, I start to see gray hair or you start to not recover. So you're interested in longevity. But now that we know that this disease, this virus that is highly transmissible, we may or may not have new therapeutics or a vaccine in a short period of time. All of a sudden now people are like really excited about maybe looking at the mechanisms of aging.
So I actually, it has not excited me at all. In fact, if anything, it makes me more frustrated because that hasn't happened. Okay. So, and in fact, we've known for a long time that influenza kills a lot of people every year. We've known this for a long time, and there has not been a policy approach to dealing with the way that the biology of aging intersects with risk of dying from influenza. We know about this for Alzheimer's disease, and there hasn't been a strategic policy approach to addressing that. So I have not expected and have not seen any real substantial policy approach to developing therapies for COVID-19 based on understanding the biology of aging. And in fact, if anything, it's been the opposite. So most people don't realize this, but the National Institute of Health, which is where most biomedical federal funding for research comes from, is made up of several institutes. And each of those institutes get their own allocations from the federal government for certain amounts of money. And most of the NIH institutes are based on individual diseases. So there's a National Institute of Cancer, Heart, Lung, and Blood Institute, Diabetes Institute, and then there's NIAID, which is the National Institute for Allergy and Infectious Disease. So when the federal government allocated several $100 million for COVID-19 dedicated funding, the vast majority of that went to NIAID, which makes sense. It is an infectious disease. Some of it went to Heart, Lung, and Blood. Some of it went to the other institutes. $0 went to the National Institute on Aging, despite the fact that age is the greatest risk factor for dying from COVID-19. So there is a disconnect at the level of policy, at least at the federal government level. And I think even among the broader scientific community, there is a disappointing level of ignorance about the importance of biological aging for things like influenza and COVID-19. And so while I and many of my colleagues immediately saw the importance of biological aging here and the great potential for reducing the impact of COVID-19 from targeting biological aging, there has not been any significant movement towards actually accomplishing that. Oh. And so, and I and others in my field have done our best to try to educate people, but that's a hard thing to do. And it goes beyond just informing people. There are, challenges, as you might imagine, to reallocating funding from one federal body to another. And people at NIAID do not want to see their federal allocations go to National Institute on Aging. So it goes beyond even what is, from a scientific perspective, what makes the most sense. There are other forces at play that make it difficult to really, from a research perspective, take the most efficient and effective strategies and apply them to something like COVID-19. It's extremely unfortunate, but that has been the case. So it's kind of funny that the way you said that you must have been excited. If anything, it's been frustrating because I really do believe that there is a lot of potential from exploring the interventions that we know about that affect biological aging in the context of COVID-19, both in terms of reducing the risk that people who are infected with COVID-19 will die, but also in terms of improving the effectiveness of a vaccine once we have a vaccine. This is another area that, there's a lot of excitement and hope pinned on development of a COVID-19 vaccine. What is not talked about, unfortunately, is the fact that the people who really, really need that vaccine, the older people who are at most risk of severe outcomes from COVID-19, are the ones where the vaccine is least likely to work because they don't have a robust immune system. And there's evidence that drugs like rapamycin that target the biology of aging can actually help with that. That if you were to give these interventions to older people, you can actually improve their ability to respond to a vaccine. Again, I don't want to say it's not being explored at all, but those strategies are being explored in extremely small studies, rare cases, whereas there is a huge amount of money that's going towards, in my opinion, research that is unlikely to have a significant impact on the pandemic.
This kind of one bug, one drug, the next blockbuster, that's kind of where it's focused. So,
yeah.
I mean, I hate to say follow the money, but it seems like there's a lot more like, Metformin is off-label at this point, Rapamycin generic, is it? I mean, so it doesn't seem like there's a lot, if you're going to invest R&D in that, what's ROI going to be versus a new vaccine that's going to be massive?
So, yeah, I mean, I think that's true. I don't think that specific example is what's driving the lack of investment in those sorts of research avenues for COVID-19.
Sure.
Because you could certainly say, well, okay, I'm not going to look at rapamycin, but I'm going to look at my own, derivative of rapamycin that is on patent and test it to see if it affects COVID-19. And that is happening again in a few small cases. Cool. But I think that from my perspective, the bigger issue is more that there is a lack of understanding and appreciation among those who allocate the federal spending for things like COVID-19, and even among the broader scientific community, that we understand enough about biological aging that we have a real shot. In fact, we know we can modulate it. There's no question about that anymore. And that by doing so, there is a very high potential that we can actually improve the immune system in ways that are going to be significant, both for enhancing a vaccine once we've got one, and also for preventing older people who are at highest risk from having their infection progress to the point where they are on a ventilator or they eventually die.
Yeah. And so these papers, like the one that you and Nir just published at the end of July, so these papers are partly is it to convince the scientific community or kind of let people that are maybe not following the research that you are, like, here's the mechanisms, here's why we should be paying attention to this? Because I know Sinclair and colleagues, I think it was end of April or early May, they published another paper, I'm going to get the title wrong, but it was something to the effect of why are elderly dying at a higher rate? And it went through all the biological aging related mechanisms that could predispose someone to severe disease.
Right.
And I thought, this is so exciting, really cool. But then you look at how many times it's referenced in relation to other COVID related articles, 3 or 4 journals. You're like, really?
Yeah. I mean, this is part of the challenge with the silos that scientists operate in, right? We all, we work in our own field. There are two actual real challenges with the way that science is fractured into specific specialties, right? One is that in general, most scientists don't know a lot about other areas. So the immunologists who study, things like influenza and coronavirus probably have heard almost nothing about biological aging. And then the second challenge is that most scientists, I think by nature, we're kind of this way. We tend to think we know a lot about things that we know nothing about. So we have strong opinions that are often misinformed about other areas of science. And so I think there is a perception among the broader scientific community, speaking in generalities now, that the biology of aging is still fairly descriptive and not particularly mechanistic. And that was true 30 years ago. That's not true today. But I think there's still this perception that people think they know something about the biology of aging, but what they think they know is wrong. And so I think those two factors of being uninformed and then also having strong opinions about things that you're uninformed about, sort of create this really challenging environment to try to educate our scientific colleagues that this is something they should be paying attention to. And so I think, the strategy that some of us have been trying to take is a combination of the kinds of papers that you were talking about, where you really dig into the mechanisms and you lay it all out and you say, aging is important for COVID-19, and here's exactly why in a very detailed way. And then there's sort of the more conceptual, which I think this falls a little bit more in that category, conceptual arguments that, COVID-19 is an infectious disease. We know that older people are at an exponentially greater risk of dying from COVID-19, and it's because their immune system, as part of the aging process, stops functioning the way it's supposed to. And we have evidence that at least some drugs that affect aging also rejuvenate the immune system, at least in mice. So maybe we should take a look at that in people and see if that might work. And then there's sort of the third strategy, which I've also dabbled in a little bit, which is to just try to go directly to the policymakers and the general public. So I published an article or an op-ed that was published in The Hill a few months ago, which is, a general audience sort of thing, just basically trying to make that same argument that I, just elucidated, but without using technical words, but again, to try and get the message across that, targeting aging biology is possible. And we have some examples where we know it works in mice. We're pretty sure it'll work at least to some extent in people. And we think that we should try that. And if it works, here's the expected impact that we might have on risk of death from COVID-19. In that Hill article, I did some sort of back of the envelope calculations. And basically, if you take what we know a drug like rapamycin can do in mice, and you say, okay, let's say it works half as well in people. What you essentially do is you bring the risk of, you basically push the risk of death from COVID-19 back 20 years. So, we know that if you go from 40 to 60, your risk of dying from COVID-19 goes up something like 15 or 20 fold. Basically, you reduce the risk of dying by 15 or 20 fold from COVID-19, which takes us from, a global pandemic to a mild flu season in terms of number of deaths. It's still a lot of deaths, but it's not this sort of situation where the entire world is gripped with fear and economies are being devastated. Obviously, it's too late to do that for COVID-19 now. We can still have an impact, but the other point that I think is important to consider is that unlike a vaccine, which is only going to work for COVID-19, and it's not even clear that it's going to work for all the strains of COVID-19 that might be out there now, because influenza doesn't, by targeting the biological mechanisms of aging, it's not this very specific one virus approach. It's basically strengthening the immune system of older people so that they will be protected against COVID-19 and influenza and bacterial infections. And in that way, it's a much more effective, I think, strategy at improving health among the most vulnerable segments of our population.
And more sustainable too, because who's to say that there's not some new superbug that's emerging that we just yet don't know about?
Yeah, that's right.
There could be something else down the road that comes, and then what are we going to do? How long can we sustain lockdown after lockdown after? It's unsustainable. So
it is, and I think also, you're right. There may be the next pandemic that we don't know about, but I think also, we've gotten used to the idea that 100-some million people are going to die every year from influenza. It doesn't have to be that way. This gets to the idea that aging is something natural that we should just accept, which I think is the way a lot of people think about aging. I think we need to get out of that mindset. I think that chronological aging, just the passage of time. I agree. I'm not a physicist, but I'm pretty sure we can't do much about that right now. Biological aging, we have the opportunity to really have an impact, and aging doesn't have to be, biological aging doesn't have to be this inevitable process where you just break down and you get frail towards the last couple of decades of your life. I'm not saying that we can cure aging, and in fact, I really don't like that phrase, but I think that we can have a significant impact on the biology of aging such that we can improve the health of people as they're getting older substantially. And maybe, again, I think it's not unreasonable to expect that at least for certain systems like the immune system, we can probably get a 20-year restoration of function, maybe more. So I think that there are real opportunities to impact aging and enhance healthy lifespan, but there is this challenge of getting people to think a different way about aging.
That it's modifiable.
That's right.
Yeah, I think, that's one of the questions I was going to ask you. I mean, if you were to ask 100 people on the street, maybe 80% of them would say, yeah, it's based on your genetics. Like my mother looked good when she was 50 or my grandmother.
Right.
I think a lot of people think it's kind of this fatalistic, deterministic kind
That's
of
right.
process. But, your work, David Sinclair's work, a lot of other folks are saying, hey, no, we can modify this. And studies have shown that with epigenetic clocks. You've shown it in dogs. So maybe let's talk about the modifiability and then kind of hone in on some of the mechanisms that you're excited about. I know when I was going to school as an undergrad, I went to Western, and it was
I went to Western too.
That's right. I saw in your bio. yeah. It was all about the free radical theory of aging, right? Yeah. And the older you get, the more free radicals you make, and that damages your DNA, and then so it goes. But now we know there's so much more that goes into it.
That's right. And I think, if you look at the history of the field, there have been a series of hypotheses for specific causes of aging that have come and gone over the years. And I don't want to say the free radical theory of aging is gone, because it's not. I think what we've learned is that it's more complicated than that. And, telomere shortening is another one that sort of gained a lot of public interest for a while, that telomeres are the ends of our chromosomes. They get shorter as you age. At some point, they get so short that makes something bad happen in the cell, and then you age. That happens. That's true, but that's not the only thing that contributes to biological aging. And so I think what we've learned is that there are multiple what people call hallmarks of aging. And that's a useful sort of framework, I think, to think about it. So these are molecular processes that happen apparently in most or all animals as we age that contribute to the declines in function and the diseases that go along with aging. And there are, depending on who you ask, somewhere between 8 and 10 hallmarks of aging. And again, those are simplifications, but you almost have to have those simplifications in order to really talk in an understandable way about the biology of aging. I really think about it more as an extremely complicated network. There are many proteins involved in the aging network that are perturbed as we get older, and some of those components of the network are particularly useful for interventions to modify the aging process. And we know about a handful of those now. And I think that there are many more interventions out there that either target the components that we know about, or that will target other components of the network that we don't know about yet, that can have significant impacts on health span and lifespan. That's a very sort of abstract way of thinking about it. And obviously I could sit down and talk about, what the specific names are of some of the parts of the network, but I also tend not to like to do that because I think there's we understand 10% of aging, and there's 90%, maybe more, that we don't understand. Wow. And so I think that by putting names on things, it's useful from a sort of categorization perspective. And the hallmarks of aging are nice because then you can explain to people that, now that we understand what these hallmarks are, if we can develop drugs that targets one of those hallmarks, that has the potential to have a positive impact on lifespan or health span. If we can develop drugs that target more than one of these hallmarks, that's even better. And that's kind of where we're at now. We've got drugs. We know about some interventions, dietary, lifestyle interventions, that seem to have effects on multiple hallmarks of aging, and in at least laboratory studies, increase lifespan and seem to delay the functional declines that go along with aging. Whether they delay everything that goes wrong with aging, I think, is still an open question. And my intuition is that the answer is probably no, that it's going to be very hard to find a single intervention that affects everything that goes wrong during aging. But what seems to be the case is that we have interventions that affect a lot of things that go wrong during aging, definitely can significantly increase lifespan, and seem to allow animals to avoid most of the major diseases of aging, and to the extent that we can tell how happy and healthy a mouse is, seem to maintain health during aging.
What's interesting about that is you're speaking about the networks, and that a drug like rapamycin or just basically reducing the activity of a central energy sensing enzyme, a kinase, mTOR, that it has a network effect that affects a lot of these different hallmarks of aging.
That's
Normally,
right.
like when you think about let's just say, well, I guess antihistamines affect histamine and have all these side effects, but it's interesting that this one molecular switch, this kinase, affects the entire network.
Yeah.
So it seems that that's like a critical target, mTOR.
It definitely is a critical target. And one of the interesting things that, has emerged, I think, from the literature in the last decade or so is that most of the major players that we know about from a genetic perspective in aging are pretty closely connected in this network that I'm talking about. So mTOR, for example, talks to AMP kinase, which is thought to be at least one of the major targets of metformin. mTOR and AMP kinase talk to sirtuins. All of those talk to the insulin signaling pathway. So there's clear connections between the genetic factors that came out of studies that were done in the field over the last 2 or 3 decades. Mm-hmm. So that's reassuring in one sense, but it also makes you wonder if does that mean that really all of these things are ultimately impinging on the same mechanisms, or that there's just some level of complexity that we don't understand yet. I think the other interesting aspect to something like mTOR is to think about, so that's the observation. The observation is that if we give rapamycin to yeast or worms or flies or mice, we know that rapamycin is an inhibitor of mTOR, and we see that it increases lifespan and appears to slow aging in all of those animals. What's the explanation, right? So why is it that you can target very specifically one enzyme and get these whole animal effects on a process as complicated as aging? And so I think for that, you almost have to turn to trying to think about, from an evolutionary perspective, how can we explain the observation that we can modify one gene and have effects on aging. And in fact, we can modify many individual genes and have effects on aging. And I think one explanation which appeals to me, and I have yet to see any data that contradicts this, is that it really comes down to this fundamental evolutionary decision that all cells and organisms have to make, which is to sense their environment and make a decision: is it a good time to reproduce, or is it not a good time to reproduce? And when you think about it from that sort of very simplistic perspective, what would be the most important components of the environment that an organism would need to consider when making this decision? Should I have babies or should I not have babies? And food is a really important one, right? If there's not a lot of food around, probably not a good idea to have babies. Whereas, if there is a lot of food around, then it makes sense to reproduce, because you then have enough nutrition for your offspring to make it to reproductive age, so that you can carry on this sort of evolutionary process. And so that's a really, the fact that is such an important decision means that there is a high selective pressure to make the right decision. So individuals who make the wrong decision at that point don't pass their genes on to the next generation. And what we've seen is that all of these major players in aging have a role in that decision process. So AMP kinase is an energy sensor in the cell. The insulin signaling pathway is a hormonal pathway that responds to nutrients in particular. And mTOR, of course, is a major nutrient sensor in the cell. And they all help the cell and the organism to make the right decision. So so then why would that affect aging? And this is where it gets to be a little bit more speculative, but I think it makes sense that, again, if you're that organism that has to make that decision, when nutrient levels are low, which is the signal for turning down mTOR, if you're not going to reproduce, the alternative strategy is to become stress resistant, right? So that you can live long enough to the point where the environment then becomes favorable when there is food around. And I think it's that stress resistant state that is associated with slower aging. And we, again, it's a correlation, but it's a correlation that comes up over and over and over again, is that animals, when we perturb them in a way that makes them stress resistant, are long-lived, and vice versa. It's not a perfect correlation. You can find situations where you can make a stress-resistant cell and it's not long-lived, but it's a pretty good correlation. And so I think that's at least part of the answer for why you can have individual genes that are conserved to play this role of regulating aging. And it's kind of a minor point, but it's also I think an important one. The important thing to note about that is that those genes were selected by evolution to make this decision. They were not selected by evolution to affect aging. The effect on aging is a byproduct, but it's an indirect byproduct. And I think this is where people tend to get confused in thinking that natural selection and evolution is regulating lifespan. It's an accident in some ways, I think, of natural selection and evolution that these genes have secondary effects on aging. That's not why they were selected to have that function. They were probably selected to have that function to, again, make this decision between fast reproduction, low stress resistance versus slow reproduction, high stress resistance. Just happens that high stress resistant state is associated with slower aging. And in long-lived animals like us, there's this idea that it's not so much that these things like mTOR accelerate aging when we're young. They might, but they're really important for development and getting us to sexual maturity. But it's the unregulated continued expression of these pathways at high levels, particularly in environments like we see in the United States, where we're sort of, you could argue, overfed, right? So our environment and our sort of dietary choices exacerbate the problem, but it's the continued expression of things like the mTOR pathway that are what really is driving aging. And that explains why you can take a drug like rapamycin in middle age, turn that off or turn it down, and then you start to uncover these benefits in the context of aging.
And so mechanistically, next scene in the movie, 21st Century Life, there's not that hormetic stress to, kind of preserve what you have in nutrient devoid conditions. So we're chronically feeding, we're constantly growing, dividing, et cetera. Are we just essentially exhausting resources at the cellular level, or is that where you tie into all these different hallmarks? Because what is it about constant growth that, I guess, mechanistically links to aging?
Right. So I think, again, this is where it starts to get complicated. The answer is that there's no one mechanism for all tissues or cell types, right? So our body is made up of lots of different types of cells. And the effect of continued expression of these pro-growth pathways in different cell types is different. So in dividing cells, your risk of developing cancer goes up dramatically the more the cells divide. I mean, it's in some ways it's simple math. There's metabolism that ties into it and damage from free radicals and all of that. But there's also just this basic mathematical principle that if you have a certain risk of developing a mutation that will cause that cell to become cancerous, with every division, the more divisions you have, the higher the risk. So in cells that divide a lot, being in that sort of chronic pro-growth state, just that alone increases your risk of developing cancer. And we know that most cancers are age associated in that way. That also potentially explains why things like intermittent fasting could have beneficial effects. It's not the whole explanation, but it's part of it, that you tamp down those pro-growth signals when you're in the fasted state.
And what cells would those be? Like, we hear that cancer of the pancreas is pretty problematic.
Yeah.
Lung cancer, like what cells are
Yeah, so the lung epithelium is pretty quickly dividing. Intestinal cells are pretty quickly turned over. Liver cells are pretty quickly turned over. So those are what are thought to be highly mitotic cell types. Yeah. In cells that are primarily post-mitotic, like most of the cells in the brain are thought to be largely post-mitotic. The heart is largely post-mitotic, not largely non-dividing. The continued expression of these pro-growth pathways certainly is associated with higher levels of oxidative stress. We know that high levels of oxidative stress arising, at least largely from metabolic processes, can cause cells to go into what's called a senescent state. That can happen in dividing cells too. And in this senescent state, those cells start to give off a pro-inflammatory set of signals. So there's a lot of evidence now. It's certainly the case that as mammals age, there is a dramatic increase in the abundance of pro-inflammatory cytokines, and this has been referred to as inflammaging, right? So it's chronic inflammation in the absence of infection that we see just as part of the aging process, and that is at least partially driven by the accumulation of these senescent cells. And my personal view, and this isn't a view that has evolved. So, I mean, full disclosure, 10 years ago, I was not really excited about the idea of inflammation, in part because I got my start working in yeast, which are single-celled organisms that don't experience inflammation. But I have certainly come to appreciate that in complicated animals, and in mammals in particular, that the increase in sterile inflammation that we see with aging, I think, is driving many of the functional declines and diseases that are associated with aging. Maybe I should say that a little bit differently. I don't know to the extent to which it is causing those diseases. I think it creates a physiology that is permissive for a lot of these diseases. And that chronic inflammatory state is driven, at least in part, by the accumulation of senescent cells. So one strategy that people are very excited about is to try to get rid of these senescent cells, turn down that chronic inflammatory state, and see what effect that has on aging. And the data so far are I think, quite compelling. At least in a mouse, if you clear these senescent cells that accumulate during aging, you see a dramatic reduction in inflammatory molecules and inflammatory signaling. You see increased lifespan, and you see functional improvements that really look a lot like the effects that we see with rapamycin. And in fact, there's emerging data now from my lab and others that these two processes are probably linked, that when we treat old mice with rapamycin, even after as short as 8 weeks, we see that it turns down this senescence-associated inflammatory response. And so my intuition is that is a big part. It's not everything, but it's a big part of the broad beneficial effects we see from rapamycin in the context of aging.
Wow, very fascinating. Just to kind of break it down a little bit for folks that hear about inflammation, Sally Smith maybe has gotten a food allergy test, for example. Yeah. So she knows like, hey, I react when I have wheat, maybe. Let's just say, make it super simple. But what you're saying here is there's these accumulation. Are they immune cells, or are they just like, let's just use the heart as an example. Right. And we know that in adipose tissue, as you gain fat, you get an infiltration of all sorts of immune cells within the adipocytes. But these the secretome that's secreted by these senescent cells that creates a sterile inflammation. So it's like your own cells are making the inflammation. It's not something that's coming from your diet. So I think a lot of people need to sort of think about this, because they hear, oh, my CRP is high, but I don't eat anything that's inflammatory. Right. But they don't realize that it could be the buildup of these senescent cells.
Or a combination, right? So I think that's exactly right. And this is a pretty active area of research. So I think that it's still being figured out exactly which cell types can become senescent, and of those, do the specific signals that are given off by, say, senescent adipocytes versus senescent fibroblasts, how different are they? So everything that I just said is a generalization that we're learning as a field, we're learning a lot more about. But it does seem to be the case that many different cell types in many different tissues can become senescent during aging, and that there is at least a core set of inflammatory, what are called cytokines, inflammatory signals that are given off by these cells that seems to be fairly general for different cell types when they become senescent. But it is also clear that there are many differences in, say, a senescent adipocyte from a senescent fibroblast that could be important for the effects of that senescent cell on surrounding tissues, and also could be important for strategies to target those different types of senescent cells to clear them, or to at least try to shut off that inflammatory response.
Super fascinating. When you hear cytokines, you think the immune system, but what we're saying is, like, in, say, cardiomyopathy, myocytes are then releasing, say, interleukin-6 or this.
So again, I think this is really being figured out. So what I think is clear is that in cell culture, so if you take a fibroblast or a myocyte out, you culture it in the laboratory. If, say, for example, you treat it with a high level of oxidative stress, you can cause that cell to become senescent and give off inflammatory cytokines. In the context of an animal or a person, I think we don't know for sure that that's what's happening. What we see is an increase in cells, I think, in almost every tissue where people have looked. There may be certain cases where it's not quite so clear, but in at least most tissues, with age, you see an increase in cells that have these molecular markers of senescence that are giving off inflammatory cytokines. Where exactly those senescent cells came from is still a little bit unclear.
Mm-hmm. Gotcha. That's super fascinating. One thing that comes to mind is exercise. And if we think about myokines, so someone might hear inflammation's bad, but if you and I go do a HIIT workout, our muscles will release interleukin-6, but it's a transient increase. And so there's this difference between this, what you called sterile inflammation, this chronic kind of smoldering inflammation, and a transient bump.
Yeah, and I'll be honest with you, I don't understand it. I mean, I think you're right, intuitively. And this also is related to, like, the idea that rapamycin or mTOR inhibition, would be a bad thing, that you should get muscle wasting because mTOR is required for muscle growth, right? Mm-hmm. But you don't see that. So I think you're absolutely right that some of these same signals and markers that we look at in the context of inflammation or aging, in the context of exercise, those markers are going the other direction. And yet there's no question that exercise is beneficial in the context of aging, at least to a certain point. And I think you're probably right. I mean, there is some evidence to support the idea that what you really need are appropriate cycles of these growth promoting versus growth inhibiting responses. And that certainly could explain, some of the observations around exercise, that people who exercise moderately but regularly are protected against diseases of aging. And I mean, I think we all know just from our own lives, they tend to age better. Yeah. So it might be this regular cycling of growth promotion in the tissues where that's beneficial. So we know it's beneficial to maintain muscle mass as you get older, but not having the sort of chronic growth promoting signaling, broadly speaking, throughout the entire body. Mm-hmm. And that's very hand wavy, but that's kind of at least the level of, given my level of the understanding of what's going on, the way I'm thinking about it now.
It's really interesting. What gets lost, and I'm glad that you're speaking out more towards the public and writing articles like in The Hill, but context tends to get lost. So I'm glad that we're kind of talking about chronic versus acute. Really interesting stuff. But getting back to the immune system, why is it that the immune system, so you talked about giving rapamycin might delay the age of the immune system or set it back about 20 years.
Yeah.
What is it about the immune system? And as I look at some of these articles, whether it's senolytic therapies, whether it's your research in animals, Steve Horvath, the Horvath clocks, they're all looking at the immune system. So why does the immune system, or is it accessible, or does it age faster than other tissue types?
Right. So I think it depends on the situation. So I think with things like the epigenetic clock, really the only reason why most of the studies have been on blood cells is because that's what's easy to get, right?
Yeah.
And what is known is that in other tissues you can create similar epigenetic clocks. So I don't think there's anything special in that context about blood cells. I think it's just that's what's easy to get especially in a longitudinal manner from the same people over time. So in the context of aging and mTOR and rapamycin, I guess the first thing I should say is I'm not an immunologist. I've tried multiple times to educate myself about immunology, and I failed miserably every time. So everything I say,
complicated, or
well, there's also a whole nomenclature that goes along with immunology it's like learning a foreign language.
Wow.
So everything I say is coming from a sort of relatively informed biologist who knows something about the biology of aging, but who's not an immunologist. Okay, so with that preface, what we know is that and I should also add that this is an area where my own appreciation of the importance of immune function and immune senescence in aging has grown dramatically over the last 5 to 10 years. Mm-hmm. I was sort of a skeptic of the idea that this was going to be, a particularly important component of aging, but now I 100% believe it. And this actually ties directly into the sort of chronic sterile inflammatory state. It's all related to the changes in the immune system that go along with aging. So what we know is that as animals get older, we've already talked about this increase in inflammatory signals. Much of that increase in inflammatory signaling is probably coming from immune cells, as you've sort of already alluded to. Probably not all of it, but a lot of it is. And one of the major consequences of that chronic inflammatory state is that the immune system is no longer able to respond appropriately to the challenges that it experiences. And there are actually, I think, a couple of consequences of this. One is you get the immune system responding inappropriately to self-antigens, and that's why we see an increase in autoimmunity with aging. And you get an inability to respond to the sort of acute challenge of influenza infection or COVID-19 infection, which explains why the elderly are at so much higher risk of severe complications from those diseases. A third really important and underappreciated consequence of the senescence of the immune system is failure to surveil cancers early on. So cancer immunosurveillance is extremely important, and it might even be the most important mechanism that we have as humans to prevent cancers before they metastasize. And with aging, there's a decrease in the immune system ability to actually do that, which is probably why we see an increase in lots of cancers, lots of different types of cancers with age. This also has important implications for, there's a lot of excitement about immune cancer therapies. There are a lot of questions in my mind about how well that's going to work in older people where you really want it to work. But that's a sort of an aside. So how does rapamycin affect the immune system? First of all, it's a little bit counterintuitive because rapamycin historically has been used or at least talked about as an immune suppressant. And that's because it's been given to organ transplant patients to prevent rejection of the transplanted organ. It's important to consider that in those patients, they're always getting true immunosuppressants. So they're taking other drugs and they're taking high doses of rapamycin. The observation in mice, and there's a little bit of data in people, is that if you take an old mouse with a or subfunctional immune system and you give it a short-term treatment with rapamycin, that mouse's immune system is then able to function as if it was a young mouse. The mechanisms for how that's working, I think, are still a little bit, a lot unknown. So certainly we know that rapamycin tamps down the sort of chronic inflammatory state. Simply reducing that sterile inflammation might be enough to allow the immune system to function more like a youthful immune system. In part, that will allow the immune stem cells that are there to actually work the way that they're supposed to. There are other effects that rapamycin has, both cell intrinsic and then systemic, that could also contribute to these changes that we see in immune function. But observationally, what is seen is that a short-term treatment with rapamycin, again, at least in mice, allows those old mice to respond to a vaccine the way a young mouse does. And I alluded to the fact that there are a couple of clinical trials now in people that tested a derivative of rapamycin. It's called Everolimus or RAD001 that showed exactly the same thing in healthy older people, that 6 weeks of treatment with this derivative of rapamycin allowed the immune system of those older people to respond better to an influenza vaccine. So at least for flu vaccine, it seems pretty clear that in both mice and people, you can boost the ability of the immune system to respond like a young immune system. What's unclear is how broad that enhanced function is going to be against other infectious diseases. A little bit of data supporting the idea that those same people who got the 6 weeks of Everolimus also had fewer respiratory tract infections over the next year. Wow. So it suggests that their immune system wasn't just primed for a vaccine response, it actually was functioning better against a variety of different types of infectious agents.
It's so impressive, just 6 weeks of therapy.
Yeah, I mean, in some ways, yeah, it is. I think, again, if you had asked people 10, 15 years ago, well, maybe 10 years ago, people were starting to figure it out. But 15 years ago, what do you think? If we gave mice or people rapamycin for 6 to 8 weeks, what would the effect be? Almost everybody would have said nothing, right? And I think that's actually been a paradigm shift in the field that stemmed, it was started with, there was a paper published in 2009 showing that you could start treating mice with rapamycin at 20 months of age. And if you started them at 20 months of age and you just continued the treatment till the end of their life, you saw significant lifespan extension and in that case, reduced rates of cancer. That was really the first demonstration where it was sort of rock solid that you could start an intervention in middle age. And 20 weeks in a mice is maybe the human equivalent of 60 years old. That you could start an intervention in middle age and get robust effects on lifespan and at least some measures of health during aging. And that, I think, certainly for me, it changed the way that I thought about interventions in the context of aging. And it was really, in my mind, that was the moment when it crystallized for me that we really have a chance to actually do this in people. It was hard for me to imagine, trying to get teenagers to start caloric restriction and stay calorically restricted for the rest of their life. That's just not going to happen. But a drug or a lifestyle intervention that could be implemented when people are 40, 50, 60, maybe even 70, and still have robust effects, that seems much more reasonable to think about. And so that was really the first paper that demonstrated that was possible. Now what we know is that at least for things like rapamycin or clearance of senescent cells, you don't even have to treat them for the rest of their life. You can get pretty strong benefits from a short-term period of treatment, even just one, 8-week to 12-week treatment in middle age, and those effects persist. So the mechanisms are still being worked out, but the observation now seems pretty clear that that's possible, and it's not even specific only for rapamycin.
That's amazing. So the dogs that you worked on, that was a few years ago, the paper that you published, the Dog Aging Project, is that it?
Yeah, so the first paper that we published was a few years ago. That was a short-term veterinary clinical trial that was actually done at a VCA veterinary clinic in Linwood, so here locally. The goal of that trial was primarily a proof of principle. First of all, that it was for safety to sort of make sure that there weren't any significant side effects over that 10-week period. But also it was sort of proof of principle that owners would participate in a study like this. So this was a study all on companion dogs living with their owners. And it was really the first, at least that I know of, the first study of its kind in healthy companion dogs. And so that was a 10-week trial, double-blind, placebo-controlled. We focused a lot on safety and monitoring side effects, but the dogs also got echocardiograms before and after the 10-week period. And that was based on 2 papers at the time. Now there are more in mice showing that 10 weeks of treatment with rapamycin is enough to see improvements in echocardiographic parameters during aging. So So that study, by I think every metric that we could have hoped for was a success. We didn't see any evidence for significant side effects. We saw some evidence for improvements in cardiac function, and that really set the stage that allowed us now to move forward to get funding to do a much larger clinical trial, which, our plan was actually to be enrolling dogs third quarter of this year. That all sort of got set back by COVID. But the infrastructure is mostly in place, and I think that we will be we'll get the first few dogs hopefully coming into the clinic towards the end of this year, and then we'll really start to enroll dogs probably February of 2021 is our target date. So that's going to be a study of up to 500 dogs, double-blind, placebo-controlled. So half the dogs will get rapamycin, half will get placebo. It will be the clinical part of that trial will be performed at 7 veterinary teaching hospitals around the United States. So it's a pretty big study. The primary endpoint for that trial is lifespan. So that's part of the reason for why we're enrolling 500 dogs, is that will give us statistical power to see a 15% effect on lifespan, if there is an effect there to see. So that's the primary goal of that study, is to answer the question, does rapamycin extend lifespan in middle-aged dogs? We'll also have secondary endpoints that look at cardiac function, cognitive function, kidney function, cancer incidence, activity. So we're monitoring a variety of age-related health measures, because our hypothesis is that if rapamycin extends lifespan in dogs, it will also delay most of the functional declines and diseases that go along with aging.
Wow. What's the inclusion kind of age range?
Right. So the dogs have to be at least 7 years old and at least 40 pounds to be included in the study. The other primary exclusion criteria is they can't have any significant pre-existing age-related condition. Because it's a study of healthy aging, we felt that it was important not to enroll dogs where they had already developed at least one age-related disease.
Mm-hmm. And they're on medication or being treated for that. So you'd want to exclude for that.
Yeah. So this is actually an interesting complicating factor is, the way the study is designed, we know that many of the dogs after enrollment will develop age-related diseases. And in fact, will reach the end of their natural life. And I mean, if you really think about it, the only possible way we could see a significant extension in lifespan from rapamycin is if many of the dogs in the study reach the end of their natural life who are on placebo. So we have already worked out, there are relatively few cases where rapamycin is contraindicated with other therapies that are common for age-related diseases in dogs. But in those cases, the dogs will come off of, we won't know if they're in placebo or rapamycin at that point, but they'll come off of whatever treatment they're getting, so that it doesn't interfere with the normal treatment that they would receive. There are, this is something that I learned having come from a very basic science background. Clinical trials are really complicated.
Oh, I can imagine.
And there's a lot of guesswork involved. And there are lots of questions that you have to think about that can impact the outcome of the clinical trial, and you just have to try to make your best decision based on the data that you have at the time. Another example is sex. So we know in mice that at any given dose of rapamycin, it seems as though the biological activity is higher in females than in males. The mechanisms aren't really understood. So, when you're designing a clinical trial in dogs, we don't have any reason to think that it's different in males and females. So do we try to only enroll females, or do we try to only enroll males? We made the decision that we weren't going to limit it based on sex, at least not male, female, because we don't have any data telling us to do that, but it could be important. The other thing that's a little bit different about dogs is dogs actually have 4 common sexes. If you really think about it, there's intact males, intact females, and sterile males and sterile females. And certainly sex hormones could interact with rapamycin. They probably interact with aging. So, how do we do that? And we just made the decision that we were going to limit enrollment to sterilized dogs. So we will have For the first study, we didn't do that, but for the long-term study, it will be all sterilized dogs, but roughly equally distributed between male and female dogs.
That makes sense. And from a dosing standpoint, is this every day, once a week?
Yeah. So the plan is that it will be 3 times a week. That is the same dosing regimen that we used in the first study. Again, this is an example of there was no way to know what the right way, if there is a right way to do it, there was no way to know what the right way to do it is. There are a couple of reasons why we went with 3 times a week rather than daily. One is that there, at the time, were a couple of ongoing clinical trials for different types of cancer in dogs, and they were using a 3-time-a-week protocol. And so we were able to get some information about the doses that they were using, the side effects that they were seeing, the frequency of side effects. And we felt pretty confident that we would not have any significant side effects with the doses that we were testing in that protocol. The other reason for not doing daily is that the 2 human clinical trials that I alluded to before with the rapamycin derivative, they actually tested a daily dose of 1 mg per day, which is lower than what you would use for organ transplant rejection, a once a week dose of 5 mgs once a week, and a once a week dose of 20 mgs once a week. And what they saw was that the side effects were, even though the dose was higher, the total amount was higher, the side effects were reduced by the once a week compared to daily. And one hypothesis is that the side effects with rapamycin and other mTOR inhibitors might be primarily driven by what are called trough levels, so the lowest level that the drug reaches in the blood and tissues, and that by spacing out the dosing, you allow the trough levels to essentially zero out. And it's again this idea of cycles, right? That you allow for cycles of mTOR inhibition followed by mTOR activation by spacing out the dosing.
Really fascinating. Now, does time of day matter in dosing?
No, nobody knows.
Okay.
Nobody knows, and we didn't feel like we had a good reason to enforce a specific time of day. We're encouraging owners to do it with breakfast simply because we want them to remember to do it, basically. And so saying do it at this time with breakfast, from a compliance standpoint, we think will hopefully make it more likely that the dogs will actually get the medication.
Yeah, a lot of dog owners have a routine. They go out for a walk and things like that. Yeah.
Right.
I mean, it's interesting. There was a study that was just published a couple of days ago, and they looked at vaccine response to for the BCG vaccine, and looked at just it was a small 30-arm study, a cohort in each group. One got the vaccine in the morning, like between 8 a.m. and 10 a.m. The other group got it in the afternoon between 4 and 6, and there was a dramatically different immunophenotype after that. So just the circadian biology of this mTOR and immune responses. I mean, it's really interesting how you have to almost think about that. Like you said, designing a study, you're like, gosh, how many variables do we control?
Absolutely. And I mean, this is, again, with the clinical trial, you can't control everything. So you kind of have to pick your poison, right? What do you think is most important? But there's no question that that the circadian rhythms and the normal feeding and fasting cycles, have profound effects on physiology. And to give you an example, in our mouse studies, we see very clear differences if we look at markers of mTOR signaling, if we look in mice that have been fasted versus mice that have just been fed, in a variety of tissues. And I mean, it's dramatic. It's not quite as big as the effect of rapamycin on mTOR signaling, but it's dramatic. And it can affect the interpretation of the data. And also, I think this is sometimes why you see differences in results from different labs, is one lab might have looked at tissues from the fasted mice, whereas another lab may have looked at tissues from the mice that were just fed. And you have dramatic differences in mTOR, which of course then has a lot of downstream effects. So it's really important, and it's something that is not, first of all, it's not often paid attention to, but secondly, even when it is, it's so complicated that it's hard to know what to do about it.
Interesting. The synergism then between, say, something like rapamycin and metformin, a lot of people that would be interested in listening to this conversation are listening to other related podcasts. I feel like there's a lot of people that are just going to their doctors. They're doing all the healthy things they can, whether it's time-restricted feeding or fasting, exercise. So they're doing low-dose Rapa or metformin. Any reason to consider something like metformin in some of your research?
So I think that there are several pieces of data in the literature that support the idea that metformin plus rapamycin is at least as good as rapamycin and might have additional benefits. So one of the side effects, I don't really think of this as a side effect, but one of the side effects of rapamycin is that you see changes in the way that animals, or in some cases people, respond to a glucose tolerance test. So glucose tolerance test is when you fast and then you give a bolus of glucose, which is usually non-physiological. It's not an amount of sugar that we would ever really experience. But what you see, so this test, what the test measures is the amount of insulin that gets produced in response to that glucose bolus, and then how fast the blood glucose level goes up and then returns to baseline. And it's that sort of area under the curve that is often used as a measure of a healthy response, a healthy glucose tolerance response. If it takes longer for the blood glucose to return to baseline, that's usually thought of as a bad thing, right? And certainly in diabetics, that's what you see. So one of the observations with chronic rapamycin treatment in mice and in some organ transplant patients taking rapamycin is that they have a poorer response to a glucose tolerance test, and in some cases in the organ transplant patients develop something that looks like diabetes. So this is talked about as a side effect of rapamycin, that you get this diabetes or pseudo-diabetes like phenotype. I actually think that's part of the same mechanism that contributes to some of the benefits that we see with rapamycin. I think that one of the things that rapamycin does is it shifts metabolism away from a primarily glucose-based metabolic state to utilizing some alternative carbon sources and perhaps ketone bodies and certainly fat metabolism, at least at some doses. And so this is my intuition that is not strongly backed by data at this point. But my guess is one of the reasons why you see an aberrant glucose tolerance response with rapamycin is that the physiology of those animals is such that they are not used to metabolizing glucose. And so when you hit them with a non-physiological amount of glucose, they're like, oh my God, what do I do? Right. And it takes a little bit to sort of respond appropriately. That's a long-winded way of saying that I'm not so convinced that the glucose tolerance side effects of rapamycin are a problem. But the observation is that if rapamycin is given with metformin, you appear to suppress that side effect. The lifespan data, this is often misunderstood or miscited by people, some of whom should know better. So the lifespan data with rapamycin plus metformin is this. Rapamycin extends lifespan, let's just say by 15% at a given dose, given a certain way. That was done in one experiment. So these lifespan experiments take 3-4 years, right? Wow. These are mice. Okay. That was done in one experiment. And then it was decided that it made sense to ask, what is the effect of metformin alone and what is the effect of metformin plus rapamycin? So that experiment was done on a different cohort of mice. The problem is there was no rapamycin alone control group. So they had, no drug, only metformin plus rapamycin. The observation from that experiment is metformin did nothing. It had no effect on lifespan. Rapamycin plus metformin extended lifespan by, let's say, 16%. So it's a little bit more than the old experiment. But because it wasn't done at the same time with the same control group, you don't know if was it really more or was it just, differences from one cohort to the other. So, my interpretation is, that's why I said that I think rapamycin plus metformin is as good as rapamycin, at least in mice. It may or may not be better. I think the human data on metformin is much more compelling than the mouse data. So we know that people with diabetes who take metformin live longer than people with diabetes who don't take metformin. No question about it. those same people seem to have reduced rates of dementia, some types of cancer, heart disease. So they seem to be protected against other age-related diseases. There's some evidence that non-diabetics taking metformin may get some of those benefits, but we just don't know at this point.
Yeah.
So if you're a diabetic, absolutely, and you can tolerate metformin, absolutely take it. If you're a non-diabetic, it might help. It probably won't hurt. Again, I think there's always this risk reward, but I think we know enough about metformin that it's probably not, going to have too many bad side effects. I do think there is some emerging evidence that, first of all, my intuition is that if you're relatively healthy and you exercise regularly, you'll get zero benefit from metformin. I might be wrong. That's just my guess. But there's also some evidence that if you take metformin and you exercise regularly, you may get less benefits from the exercise. So there may be this offsetting effect of combining metformin with exercise. That, again, it's far too early to say that with any level of confidence, but that evidence is about as good as the evidence from the preclinical studies that metformin affects aging, right? So I would sort of put those things on par. And so, from my personal perspective, I'm not taking metformin. I don't have diabetes. I'm pretty healthy for my age. I'm pretty active. I don't think that it would give me much additional benefit on top of, just living a healthy lifestyle.
Mm-hmm. It's interesting, that using it with exercise and the counter benefits, because how it affects the mitochondria and things like that. So I'll take just 500 milligrams of berberine on days that I don't exercise, just for fun, just to experiment and see. And I noticed that my ketones go up quicker and things like that. But going back to that amazing riff where you talked about kind of that metabolic memory, I think a lot of physicians don't really talk about that. And so if you're doing a ketogenic diet, because maybe you have pre-diabetes, you go to your family care doc, they're like, well, your diabetes is worse. You're like, well, wait, you've been relying on and grooving this metabolic pathway to oxidize fat. So of course, when you throw in a 77 gram of dextrose, you're going to look.
I think that's exactly right. So I've had that exact same thought, and I've never tried to look at the clinical data in people, and I don't know if that data even exists, but that is exactly my prediction for what would happen. I'm pretty. I have a pretty positive view of ketogenic diets in general. Mm-hmm. I think, unfortunately, that word, again, has sort of been misappropriated in a lot of ways. All a ketogenic diet means is that you're eating a diet where your body is producing ketone bodies. Mm-hmm. And that can happen many different ways. Unfortunately, I think the ketogenic diet, the way that it has reached the mainstream media, has gotten sort of a bad rap, which happens. No, but I think you're exactly right. My intuition is that if you were practicing a ketogenic diet for any period of time and you went and took a glucose tolerance test, you would fail miserably because it's the same thing that I was talking about in the mice. Your body's going to be like, what the hell is going on? But that doesn't mean that you're in an unhealthy metabolic state. In fact, it probably means the opposite.
Mm-hmm. It's really fascinating, but something we need to talk more about. But I want to be mindful of your time, and maybe if we could just kind of finish up on autophagy. We kind of talked about cellular senescence and things like that. One of the main mechanisms that inhibiting mTOR and driving up AMPK, using whether it's fasting, exercise, or the drugs that we've been talking about, is enhancing autophagy. Right. We kind of talked about the hallmarks of aging. When it comes to all these networks and so forth, to me, autophagy seems like it's all the rage. It's kind of like where keto was a few years ago. Like everyone's talking about it and fasting for autophagy. But what's interesting, I want to hear your thoughts about it. But since we've been talking about exercise, there was a few studies that have looked at least at the skeletal muscle, that when exercisers have the autophagy initiation proteins are more dense and microRNA is higher, but also at the onset of fasting, trained athletes compared to sedentary controls after 72 hours have higher levels of autophagy initiation protein. So it seems that, again, this lifestyle really matters. Yep. So I guess there's kind of two things. What gets you excited about autophagy, or are you? And is there kind of a flavor differences based upon someone's, exercise or...
Right. So, so I guess I would start by saying that autophagy is one important mechanism that the cell has for repairing damage that has accumulated over time. I absolutely think it's important. I think that in some ways it has gotten, like you said, popularized to the point that there's not a real understanding of what autophagy is doing and what it means when we measure some of these things. So I think one of the challenges in the field of autophagy, which I'm sort of peripherally associated with, is what we mean when we talk about autophagy. So autophagy, in a very sort of simplistic way, is a cellular process to accumulate and degrade various types of molecules. Sometimes that means damaged molecules that have accumulated, because of reactive oxygen species or for other reasons. And so it's sort of thought of as a recycling center for the cell, right? Where you can take old stuff, you break it down, and then you can use that to make new stuff. And that's one important thing that autophagy does. But autophagy does other stuff too. And so, you were talking about the autophagy initiating proteins. The ways that people have measured autophagy and what they mean when they talk about autophagy is very context dependent. And I think there's been a lot of confusion about when we make these measurements, what does it actually mean? Mm-hmm. Does an increase in autophagy initiating proteins really mean that there's an increase in that sort of final degradation? And in many cases, it doesn't. And in fact, in many cases, what we see is that turning up, so high levels of autophagy are often associated with healthier aging and lifespan. That's been seen in laboratory animals. High levels of autophagy, at least by some measures, are also associated with many pathological conditions. So turning up autophagy, depending on the context, can be beneficial or potentially detrimental. And so I try not to talk about autophagy as if it's this, universally good thing, right? And I think there is an idea that many people are coming around to, that again, thinking about these cycles of growth versus high growth versus low growth, that the same thing's probably true with autophagy. And in fact, when we actually think about it, that makes sense, because autophagy is one of the readouts of low growth signaling. So again, let's just take a step back to that sort of fundamental evolutionary question that I was talking about. When there isn't much food around, right, that's a good time to turn down mTOR, shut down cell division, shut down reproduction. That's also a good time to turn up a recycling center if you have one. And so this is all tied in to nutrient availability, pro-growth mTOR signaling. You turn down pro-growth mTOR signaling, that turns up autophagy. But that's not sustainable forever. You can't have chronic high levels of degradation, or you're going to degrade everything in the cell, and that's going to be a bad situation. So probably what you need are cycles of productive autophagy. And when I talk about autophagy in the context of good autophagy, I generally think of productive autophagy, where you're actually initiating, progressing, and completing that sort of degradation or recycling process. Mm-hmm. And you need to get to that completion point in order for it to be effective. And so that's where I think these cycles of activation of autophagy and then turning off autophagy and then turning it back on are probably important. Mm. And again, one of the consequences of this sort of chronic over-nutrition state that many people find themselves in is you don't get that appropriate cycling, right? You're always in this grow, which is always inhibiting autophagy. I think there's some evidence to support that.
Mm-hmm. So this kind of, there's a few lay press books that come to mind and basically characterized everything you just said. Eat, stop, eat. Yeah. So going through this period of, because some people get excited about autophagy. So they're like, I'm doing a 21-day fast. Yeah. You're like, well, at that point, you're probably going to degrade skeletal muscle and this and that. But it's like, well, maybe a 3-day fast once a quarter or once a month if you're obese or this or that, to have this natural cycle.
Yeah. And again, I think this is all, so I think that we should be honest with ourselves that all of these things are guesses. Some of them are based on more data than others. But I don't, at least personally, first of all, I don't think there is any dietary strategy that will work for everybody. I think that people are obviously genetically unique and environmentally unique. And I don't think there's any one sort of broad dietary strategy, whether it's intermittent fasting or ketogenic diet or whatever. I don't think there's anything that will work for everybody or work the same way for everybody. And unfortunately, I think, one of the things that I've seen happen in the aging field is a lot of people who are scientists in the aging field who have worked on dietary interventions have popularized that, which is okay, but what sometimes gets lost is the fact that they're guessing that these things are going to work for people. Yeah. And I have seen, a trend where scientists in the field have become diet gurus, and I am a little bit concerned about that. Mm-hmm. So having said that, I do think, if again, it's a guess, I do think that for many people, particularly people who are overfed, overweight, I think that some of these dietary strategies like intermittent fasting or a fasting mimicking diet are very likely to be beneficial. And again, it's kind of like metformin. Probably not going to have too much in the way of significant long-term side effects. A 21-day fast seems pretty extreme to me. Mm-hmm. Certainly, there are individuals who would be harmed by a 21-day fast. There may be some people where it's beneficial.
Right.
So I think, my personal perspective is to be a little bit pragmatic about this. And certainly, I'm, as you may have picked up on, I'm hesitant to personally say anybody should do X, right? Mm-hmm. Because I don't feel like we know enough to really make those kinds of recommendations for everybody. And I do think, from my own personal experience, I think that to some extent, you need to find what works for you. And again, I don't think there's any one approach that's going to work for everybody.
Mm-hmm. Along those lines, what approach will you, maybe in your 60s or 70s, what would you consider first, like doing more fasting or Rapamycin or Rapalog? What are you going to gravitate towards first personally?
Right. So I think one of the things I've learned about myself is fasting doesn't work for me. I don't like it. And I'm not saying it wouldn't be beneficial for me, but it's not something that I am likely to ever adopt as a mechanism that I use, right? Yeah. One of the things that I found, again, this is me personally, so I'm not making a recommendation to anybody. One of the things that I found works really well for me is a low carb diet. And it is probably ketogenic. I don't measure my ketone bodies, but given the amount of carbs that I eat, most likely I am often in ketosis. And I'm not, dogmatic about it, right? But it works well for me. I don't get hungry. I feel healthy. I have lots of energy. My exercise routines are great, so it works for me. My intuition is that that probably most Americans eat too many simple carbs. And I realize that's not like a profound statement. I think a lot of people realize that, but I do think a low carb diet will probably work for a lot of people, but it won't work for everybody. Maybe some people will do better on intermittent fasting. So that's what I naturally have gravitated towards. I'll be 50 in February, right? So that's sort of what I've gravitated towards, in my own last several years. That, and I think finding some type of exercise that works for you to do regularly, where it's not a chore that you can enjoy, is really important. I don't, and again, this is not profound. I think we know that exercise in general is good for people, but I think what's hard for most people is to find something that they can do regularly and stick with. Yeah. And, sure, maybe resistance exercise is a little bit better than endurance exercise, whatever. My view is to some extent, it doesn't matter. Get out and do something, right?
As long as you're consistent with it.
Yeah, as long as you keep at it. And if you want to change what you're doing, maybe you want to hike for a few years, and then you want to bike ride for a few years. And I think the most important thing for most people is just to get out and be active on a relatively regular basis. And then the question of, do I take metformin? Do I take rapamycin? Do I take something else? Right now, I'm not. I certainly, if I was going to take one thing, I would take rapamycin. That's what I think the data is best supporting at this point for most likely to have effects. Like I said, I think metformin will work for people who are in a compromised metabolic state for sure. It might be beneficial for people who are not in a compromised metabolic state. But I think you can get the same benefits from exercise.
Yeah, I'm with you. So what are you doing for exercise? We kind of emailed about hiking and things like that. What's your exercise look like?
So I mean, around here, one of the nice things is that even though it's gray and rainy for a lot of the year, you can always get outside. So we do a lot of hiking, and when it's not gray and rainy, we do skiing in ski season. I do resistance exercise a few times a week, 2 to 3 times a week, depending on the week. So I try to mix it up, but I'll go out and shoot baskets with my son and things like that. And, it's just do different things, but try to be active on a regular basis. I personally, for me, I think having some components of resistance training is really important.
Yeah.
I think, we've learned that maintaining muscle mass when you get in your 60s and 70s and 80s is really important. And so I think that resistance training can help with that. And, I'm not saying that it's too late to start in your 60s, but I think if you have a history of doing it, then it's easier to continue to maintain muscle mass.
Yeah, you have this foundation from which, and it becomes part of your life.
That's right. Yeah.
So I think it's really important. So your dog aging project, are you still recruiting? Do you need people to?
Absolutely. Okay. So let me just give a little background on the dog aging project. So there are, broadly speaking, 2 parts to the project. The Rapamycin trial, which we've already talked briefly about, and then what we call a longitudinal study of aging, which is observational. And that's the much larger in terms of number of dogs component of the project. So the idea there is really just to follow companion dogs over their lives, try to figure out what are the most important genetic and environmental factors that influence healthy aging in dogs. And so that study is unlimited in the sense of number of people that can participate. There is the largest group, which we call the Dog Aging Project PAC, which involves people going to the website, www.dogagingproject.org. You should go and nominate your dog to be in the Dog Aging Project. So the Dog Aging Project PAC involves going to the website. You fill out a very short form telling us a little bit about your dog, and then you'll be invited to complete what is called the Health and Life Experiences Survey. That's a fairly extensive survey that asks you lots of questions about your dog. Fortunately, a lot of people like to talk about their dogs, and so they're happy to tell us everything that we want to know about their dog. And it asks things that you might expect, like, what kind of food is your dog getting? How much time does your dog spend outside? When it's outside, is it on the grass? Is it on cement? Things like that. It also probes deeper into some of the environmental variables that people might not think about as being important. Well, we also, based on the zip code, will get information about the air quality and water quality that dog experiences. So there's a survey, and then we ask all of the owners in the pack to contact their veterinarian and have their dog's electronic veterinary medical records sent to us.
Oh, cool.
In which case we will extract data from the veterinary medical records. And then a subset of people who are in the pack will be invited to participate in more intensively studied groups of dogs. So there's the foundation cohort, which right now is set to be 8,500 dogs. We're hoping to expand it. All of those dogs will get their genome sequenced. So we'll get DNA information on those dogs, which will allow us to do the genetic mapping. And then 1,000 dogs will be selected for what we call the precision group. Those dogs annually, in addition to DNA sequencing, will have their microbiome studied, their blood metabolome, so all of the metabolites in the blood will be analyzed, and then they'll get regular veterinary exams, and then we'll also look at their epigenome. So that Horvath sort of method of the epigenetic clock, we'll be studying that in dogs. So that'll be 1,000 dogs. Again, that's what we're funded for now. Our hope is that we can expand that. So we are absolutely hoping that additional owners will sign their dogs up to participate. We have not filled any of the dogs for the Rapamycin trial, so absolutely, we have people who've already nominated their dogs, but we certainly are encouraging people to continue to, if they're interested in participating, to to nominate their dogs to be in the Rapamycin trial, which we call Triad, for test of rapamycin in aging dogs. So we already talked about the criteria for that. The dogs have to be at least 7 years old, at least 40 pounds. The owners also have to live within driving distance of one of the partner veterinary teaching hospitals. So as I mentioned, there are 7 VTHs around the United States. One is Washington State. So, it's not super convenient for people in the Seattle area to bring their dog to Wazoo twice a year, but some people are willing to do it. So owners have to be willing to bring their dog to one of the partner VTHs twice a year for the 3-year period of the study. But if you're interested and you're willing to do that, then definitely we're encouraging people to continue to participate.
Yeah. I mean, people love their dogs and take so much care. I mean, sometimes they take better care of their dogs than they do themselves.
Absolutely. Yeah. I mean, there is, I don't know how good the data is, but it's at least anecdotal that dog owners are more likely to give the dog's medication than they are to take their own medication. So.
That's funny. That's awesome. So I'll put that in the show notes, and I think that's really cool because then you can extrapolate not only to help dogs and service dogs and all that, but hopefully that data can translate into more human clinical
Absolutely.
trials.
I mean, it's both, right? Yeah. So it's both about the dogs and about what we're going to learn about human aging. And I view those as equally important goals of the project.
That's awesome. Well, Matt, thanks so much for coming out here, coming on the show, sharing amazing research and science that I'll put in the show notes. And keep up the great work. I love following some of the articles that you write, and I'm glad that you're writing more kind of layman friendly articles too, because I think the science needs to get out there.
Yeah, I agree. I mean, I think it's immensely important that we in the field of what we call geroscience or aging biology continue to try to explain to non-scientists why this is important and relevant.